So just to introduce our first plenary speaker, Pietro Cicuta is the head of biological and soft systems at the Cavendish Lab at the University of Cambridge. Pietro, are you there at all and ready to screen share?
I am, thank you, yes.
Brilliant. So his research focuses on imaging, microfluidic devices, and addressing really a range of questions on biological and soft matter physics. So we're really happy and delighted to have you at the conference, Pietro, and over to you.
Thank you very much, Josie. Can I just check, should I finish at 1:40 for 5 minutes questions or something different?
Yes, that'd be great if you've got a few minutes for questions at the end, but we can also ask them in the panel. So
okay.
Feel free to use the time as you want.
Thank you very much. Can you see my...
Yep, we can see it. It's not full screen yet, but we can still see your screen.
Okay, now it's full screen, I think. Okay, well, what a pleasure talking at this workshop. Thank you, Mia, thank you, Josie, Toby, everybody who's kind of organized it, made it run, invited me to talk. I hope not to let you down. There's not very much intelligence in my talk, but I will appeal to all of your intelligence because I will tell you kind of a story that has many sides, and I hope it makes some sense, and you can ask me questions at the end. So in terms of scale, I will be talking about microscopic things. I sort of, in that sense, perhaps follow on from Stoyan and from a few of the other talks. I will talk about this organelle, so a little piece that is present in many of the cells in various organisms called the motile cilia. And the two videos you see here, I'll show you more as we go along, are examples of these filaments, and they are driven by molecular motors and continuously beat. By beating, they move fluid. This movement of fluid has many consequences in biology and physiology, and it's a way in which many parts of a human system interface with each other between physical ways, electrical signals, and all kinds of quite complicated things. I'll tell you what we study in my lab about this in physics. So this is the work of many people over about 15 years. The people in red are those whose contribution is most present in the talk today. I just highlight the unpublished stuff has to do with using cilia as sort of computational models, and this comes at the end of the talk. And that has been the work of two final year project students in physics, David Liu and Billy Meadowcroft. So I have to introduce the concept of synchronization. Maybe many of you know about it or can imagine what it is. And one way to think about it is in the context of these experiments with metronomes, which if you Google and go on YouTube for metronome videos, you find many examples. This, I think, is the most beautiful. It has many metronomes. So the metronomes are set in motion. I will speed up a little bit because the whole thing takes 4 or 5 minutes. So they're set in motion with random phase. I'll show you how that happens. And then by waiting a few minutes, something quite interesting happens to these metronomes. Oops. So they're set in motion at random phase. Very gradually, over a few minutes, by themselves, they spontaneously start sort of ticking together, a little bit like the old days of Communist Party big congresses in sort of the Soviet Union. So this only happens if the metronomes are set to kind of to have the same periods or periods that are very close to each other, and that's one important aspect. They also sort of feel each other, in this case by a mechanical coupling, because they are all set on the same board. This is an ironing board, and the board has a little bit of vibration, and that's how they sort of can couple and transmit forces to each other. Another example that sort of I think primes you to understand what I want to say later is the famous sort of Mexican wave, which people sometimes do in big stadia. So once this wave is set in motion, sort of you stand up if your neighbor has stood up, and then you sit down again, and it gives rise to this beautiful sort of emergent property. You have a local rule, just standing up and sitting down, but collectively it creates a traveling wave which can go kind of around the stadium. So there is a theme in my talk, which other speakers yesterday and today also alluded to, the connection between simple local rules and complex emergent behavior. And sort of wave that comes from a local rule that doesn't even know what a wave is an example of that sort of very interesting behavior on a different scale. So now let's zoom in. This is the surface of a single cell organism called Paramecium. It swims around in freshwater ponds. This particular movie is a very famous movie from a Japanese lab, and it shows very nicely in slow motion what happens on the surface of this cell. So you see that there are many filaments. It's a dense carpet of filaments, and they each beat. Each one is doing a periodic motion, which is very similar to the filaments just that neighbor it. But there's a phase difference, and so collectively this is giving rise to a wave. And again, much as like the Mexican wave example, each filament doesn't really know that it has to do a wave. And this is an organism that doesn't have a neural system. It's all anchored in the same cell. So presumably the cell has one single environment. There are no sort of waves of calcium or complicated other things. So the emergence of this traveling wave must have to do simply with simpler sort of mechanical coupling and a combination of local rules giving. I'm sorry if the videos are running very slow. Yeah, I can see that on my second screen. Well, the real ones are just a little bit faster. They give you the idea of a traveling wave of cilia beats. So these cilia typically have what's known as a power stroke. So they move in one direction, kind of extended, and that way they push the fluid. So this is happening. It could be in water, or it could be in a viscoelastic material such as a mucus in other scenarios. The liquid is pushed, and then these filaments bend and go back. By bending, they then have a lower drag coefficient, and they push fluid less as they go back. And that way, sort of there's a net force and a net momentum transfer into the fluid. And so a filament can push fluid, and this allows either fluid flows across a surface if the surface is full of cilia, or it allows an organism to swim if the organism is free, like a Paramecium or many others that use this machinery for swimming. The cilia are all typically beating in a plane, and often in a physiological situation, these planes are all aligned so that you can really sort of get sort of additive properties out of all the cilia together. There are many open questions, and some of them have to do with how the cilia themselves work, so inside the filament, and other questions have to do with how they come together and beat. Apologies, I see this video is also beating quite slowly. What you should be seeing if it was transmitting better is that in this situation, these two black filaments are beating in phase, a bit like a breaststroke. Whereas in this other situation, the two filaments are out of phase. So these two movies are an algae called Chlamydomonas, which is used as a model for beating cilia. This Chlamydomonas is here. You can see my mouse a little bit on the algae cell. It's held in a micropipette. So normally this algae would swim by beating the cilia in this way, but it's held fixed. And when one can study in this experimental situation, when it beats its flagella in phase, in breaststroke, and to go straight, and when it beats them in out of phase, which allows it to turn and change direction. So this is a beautiful model system for much biology, but also for these sort of more physicsy questions, i.e., sort of what forces are transmitted between one filament and another filament. So one of the forces that dominates this coupling is the hydrodynamics. So each filament is pushing fluid, and that sets up a velocity field in the fluid, which any other filament in the vicinity will pick up. So in my lab, we've discovered lots of evidence about the importance of this hydrodynamic effect, and likewise in Ray Goldstein's lab in DAMPT here in Cambridge. There's also evidence that there are some mechanical forces in the cytoskeleton in some situations. So it's perhaps not purely hydrodynamics, but certainly hydrodynamics is enough to create lots of coupling and emergent behavior, as I will show you also in other experiments. Down here, what you see is the surface of a mouse, but this is typical of all mammals. You have in green are stained the cilia, which are about 5 microns long, and below that in this tissue section are cells that make mucus. So this happens in the airways, and it's what allows us to keep the airways clean of dust and of many bacteria and pathogens. And for example, in the COVID virus, the virus arrives and it sticks to the cilia and it stops them from beating, which is probably one reason why it then is able to reach the bottom of the lungs and create infections there. So, okay, so in my lab, we studied the biology and what goes wrong and the diseases, but that's not really the topic of today. But I wanted to tell you that these cilia in the human being, they are essential in the airways. They're also essential in the brain. In the brain, this was a beautiful paper by a Max Planck Institute in Göttingen from a couple of years ago. They managed to study live mice and image the flows driven by cilia in the brain. So carpets of cilia drive the cerebrospinal fluid, and this transports nutrients, but it also transports signaling factors from one part of the brain to another. So we know how complicated the brain is already on the just neuron connections. And then you have to imagine that there's also this sort of physical connection and physical stuff that is happening on the brain. Very complicated. So I've talked about most of cilia. This is what they look like if you take an electron microscope and zoom inside and take a section. There are microtubules, so quite rigid filaments of the protein tubulin connected by molecular motors that are sliding these filaments one with respect to the other, and this results in a net sort of bending. So this is itself a very interesting emergent property, how the motors come together to act cooperatively. Several hundreds of molecular motors are all the time attaching, exerting a force and detaching, and to make a periodic beat, that power stroke and recovery stroke that I described before. In different organisms, the structure of the number of tubules, tubulin filaments and the motors is almost exactly the same, but what can change is the length of the filament and the energy that flows in and other subtle things that result in the overall shape of the beat being different. And then that can lead to quite different emergent properties when you have two filaments or more than two filaments. Okay, so I should say these filaments beat at between 10 and 50 hertz, depending on which one you're looking at, if it's the algae or the airway or the brain of a mammal. They can be anywhere between just below 10 microns and up to 30 microns. And their diameter is 250 nanometers, and that's fairly conserved across kind of all the eukaryotes. So these are a few more videos, a bit better. These are healthy airway cells, and each cell has several hundred filaments, and they are beating, and you see kind of nice flexible beats. And this flexible beat is important in establishing coordination and waves. When things go wrong, this is a genetic class of diseases where the structure of the cilium is not quite right, and the filaments are more rigid, and also this ends up not creating a traveling wave. And so people with this disease have problems keeping the lungs healthy of infections. Okay, so I'll show you one experiment here where we studied the influence of an external flow on the beating of cilia, because that links with what I want to tell you afterwards, which will lead towards model systems. If you're curious about what I'm saying, sort of our work until a few years ago is in a long review here, and then I'll give you more references about recent work. So we did an experiment where we set up a periodic external flow using a cap on top of a layer of cells that have cilia, and we were imaging everything in an optical microscope at a high enough frequency that we could see the cilia beat. And this creates a situation which many of you are quite expert in dynamical systems from the talks I've heard, and you know about the Arnold tongue. So if you have a driven oscillating system, you can come with an external signal, and if your signal is of a similar frequency, it might be able to kind of lock the phase. Then that locking is represented here in black, and it means that you're clocking and controlling the phase of your system. Which so if we do this in this situation by changing the velocity of our external flow, if the applied velocity is very low, then we don't have control of our cilium. But if the applied velocity is strong enough, then we're exerting a strong force, and we can force the cilium to follow our external signal, even if we move away from the intrinsic beating frequency. So that creates these typical V's, which are known as Arnold tongues. So this is a typical phenomenon that you get together with synchronizing systems. The other graphs are not very important. We studied the effect of number of cilia, density of cilia, and things like that. So we created these carpets of cilia, but they're not as dense as they would be in physiological situation. So what we see here are individual cilia. Again, sorry, this is very jerky the way you're viewing it. What you should see are, again, kind of quite smooth beatings. And as we change the applied velocity, we can get several cilia on the same tissue to lock to our external drive. So in order to sort of simplify this system and go towards kind of something where we can do physics and almost engineering, one approach is to take the very complicated shapes of the cilium, which then become sort of an effect on the fluid flow around the cilium, and reduce it to just one center of force, which we call the center of drag. So we can do that from these experiments by segmenting the shapes of the cilia at each frame, and then imagining that the cilium is made of rods, and each rod is sort of creating a contribution to the fluid flow. And then we add them all up into just a one kind of sphere, one small sphere, which we move along the center of drag of the filament. If we do that, we can reduce that very complex kind of flexible filament to a sphere and to a trajectory and to a velocity along that trajectory. And sort of all the biological systems that we've in some of our work we've done the job of kind of collecting data from as diverse systems as we could. They can be reduced to this sort of unified, more simplified framework of trajectory and velocity, and then we can study how these spheres along trajectories with the right properties synchronize or don't synchronize. And at this stage, we only have hydrodynamic interactions. So we drive the spheres along trajectories, and each sphere sets up a velocity field. And we know how another sphere picks it up by something called the Oseen tensor, which basically says that the fluid flow has the velocity of your driving sphere, but it decays like 1 over the distance, or 1 over the distance cubed if you're close to a surface. And then the other sphere will pick this up as a force. So each sphere is going around its trajectory with by itself, it would have a well-defined period and a given track, but a free phase. But if they are also exerting small forces on each other, then it's possible that these phases will lock, which is the synchronization that I've been talking about before. So they might lock in phase, they might lock in antiphase, or they might lock with a small phase difference. And if there is a small phase difference and you have several of these spheres sort of perhaps in a line, then that would be the situation where you get a traveling wave along the line. Okay, so these are the papers that I'm sort of very kind of presenting in a sort of fairly superficial way. This idea of the simple sphere and studying synchronization of spheres can be also the basis of experiments. And you can trap small spheres in optical traps, which we call optical tweezers, and you can push them around by moving a focused laser beam. And you can trap several spheres because you can time share the beam or have several beams each holding one sphere. So we time share the beam, and we can trap up to a dozen spheres. So this allows to do an experiment which is basically moving the colloidal particle along a trajectory. It could be a circle, or it could be something more interesting like a bean shape, like I showed before, and it might have a modulation of velocity at particular angles. And these parameters can be matched to some biology. So we're sort of keeping some of the biological detail by representing it as a particular trajectory. So at a high frame rate, we determine where is the colloidal particle, and we put the optical trap just a little bit displaced from it in order to exert a force. And we do that kind of quickly with a feedback loop that goes through image analysis and a computer in order that we can drive the colloid particle on a precise trajectory, but leaving the phase free. So we don't just want to move the colloid in a circle with a phase that is imposed from the outside. The phase has to be free so that we can study synchronization. So this feedback and just sort of applying a force allows us to have a system that is able to synchronize or not synchronize. We calibrate a lot so that we can drive colloid. This one is fairly smooth. This colloid is going smoothly around a circle. And when we put two in our system, these two have now synchronized to go in phase. We're not forcing it ourselves. Each feedback is being done on each colloid separately. They have ended up in phase because they are transmitting a little bit of force one onto the other. So this gave rise to quite interesting physics. We have several papers now. One aspect is we can control whether they go in phase or not in phase by tuning the harmonic well to make it more rigid, like a railway track, or more floppy, so that it allows radial fluctuations as it goes round. And also, very importantly, we can modulate the velocity around the trajectory. And we've learned how to make things go in phase or out of phase by small modulations of whether we go quickly or slowly at different places along the trajectory. So this is an experiment where we, as a function of the angle on that circle, we just accelerate or decelerate a little bit, and we can go sort of, we can have almost like arbitrary phase locking at different phases between two colloids. And working with Ramin Golestanian, who's now in Göttingen, and Naria Uchida in Japan, well, they were also able to kind of create a theory that has the hydrodynamics and has the properties of the drive in order to create an effective, essentially a potential that controls the phase difference. So there's also a lot of theoretical background to what type of phase locking we would expect in this class of models. There's an even simpler model. What I've described so far is called the rotor. It's called the rotor model. But there's an even simpler model that allows you to study both experimentally and theoretically this type of synchronization. It's called the rower model. So in the rower model, we again reduce just to a bead, but then that bead is just moved along a segment, left and right, and we drive it to the right until it reaches a limiting position, at which point we drive it to the left. And then when it reaches a limiting position on the left, we drive it to the right. So again, like before, we're applying a force, and in a way that this oscillation is left to be phase free, because we're setting the average period and we're setting the amplitude, but we're not setting the phase. So if one is in isolation, it will have will go back and forth with its own phase, and if we have several, they may or may not phase lock. And here again, we can keep the biological detail in the shape of the drive that we push. We push right, for example, with a constant force, or with a force that increases, or a force that decreases. These are sort of the three things we can do, and that sets up again an arbitrary phase locking between two or more beads. So if we have several, we set them going. Okay, sorry, I didn't link this properly. It sets up sort of a collective dynamics, and we've done work based on the Oseen tensor to predict sort of what sort of collective dynamics we get from arrays. But now, in the last few minutes, if you follow me so far, what I've shown you is sort of the emergent dynamics that happens because of the physical interactions. Now, I want to show you how we can perhaps try to harness this into actual sort of more kind of computational modules. So there's been a lot of work on sort of using physical systems to create things like logic gates. This is just sort of an overview of references that I found very interesting. There's been work on sort of chemical systems using enzymes or simple chemical reactions. And it's work that I think started in this 1993 paper, I think is the key reference, the oldest reference that I could find, but it's still very much active today. There's a lot of work using DNA nanostructures, which offer a lot more flexibility. But by and large, if you're using chemistry, you're going to have kind of one-pot computations. You're going to have to set up your reagents. They then sort of do all the reactions. You'll come up with a result, but it's a little bit difficult to imagine sort of a continuous flow, except if you go sort of droplet microfluidics. But those are still sort of one-pot computations. Biology also offers many examples of computation. The most famous gene regulatory circuit is perhaps the lac operon. It's an AND gate, essentially. Two conditions need to be satisfied together in order for a bacterium cell to start producing a certain important enzyme. This is well understood, very well understood from the biochemistry and the statistical mechanics, and it's a really beautiful textbook example. But this is slow. This is something that can happen in a 1 micron bacterium, 1 micron cube bacterium, but it's very slow because it's gene expression. So it could take half an hour for this to actually take place. There's very sophisticated cell behavior, not at the gene level, but at the protein interaction level, which is often also called signaling pathways. And this is a great paper from a few years ago, where it was shown that immune system cells are able to filter signals based on the time domain. So they're almost doing kind of FM radio decryption. And so the same level of signal, but coming in at different rates, is interpreted and sort of fed into different results inside the cell. And this is important sort of in immune response. So, but again, fairly slow. So the timescales of these different things are again sort of then on the scale of gene expression, so half an hour or more. But very sophisticated computation with even examples of filtering that have come from evolution. Okay, so what have we done? So this is back to our very simple system of beads. We have beads in a linear chain, but now we're trying to set up chains that have sort of interesting properties, and we put a signal on an external signal on one of the beads. So I showed you in the examples on the cilia, we can put fluid flows. So now there's a fluid flow that hits one cilia, one bead, and we want to see how this information propagates along the chain. We can have the chain sort of with equal spacing, but if we want sort of asymmetric information transfer, one way is to make an increasing distance between the beads along the chain. And that, very interestingly, to me at least, sort of starts to give sort of a diode behavior. If you have a chain with increasing distances, it becomes very different whether you put the signal on one end or on the other end. Basically, you get information transfer only in one direction. By information transfer, I mean one of many things. You can look at the coherence of one bead to the other, the phase locking, or the coherence to this external signal that we're putting in, that they're all broadly equivalent. Once we have the diode, we can put sort of these diode chains together. And we've demonstrated, but not yet published, sort of a whole complete set of logic gates. So this is an example, 2 diodes that we've set up at 90 degrees, acting on a single bead here, can be made to be an OR gate or an AND gate, depending on the external frequencies that we add up on the powerful ends of the diode. So there's not yet sort of the non-linearity I would hope. We're still sort of optimizing this to really have a very strong AND behavior. But it's the beginning to me of using kind of these systems as modules. Okay, so I've taken you on a long tour that had a little bit of introductions to synchronization, the concept that we have these motile cilia, they are hydrodynamically interacting in many situations. The motile cilia have 100 questions and interesting behaviors. They're fascinating in developmental biology as well. I only touched on some of the things where they play important roles. I then introduced you to simple models of these motile cilia and emergent dynamics that can happen in arrays of them. In particular sort of arrangements, we can make them transfer information anisotropically, which I think I'm allowed to call a diode. And I think that because of the nature of the interactions, which can be made quite local, these diodes are kind of scalable, and they should be sort of quite good modules. Because the Cilium beats at 10 hertz, it's not as fast as a silicon computer, of course. It's not nearly as fast, but it's faster than anything that does a gene expression or chemistry. So for the future, kind of how much can we push this? I don't know. We also need sort of memory units if we really want to do computation, but we have ideas on how to make those. And sort of, can we then at some point move back from these experiments that we do in the computer with models to sort of actual physical sort of arrays of algae or cilia kind of positions on surfaces, which would be a physical realization. That would be a challenging experiment, but really fascinating. And so this is a sort of, well, last year was Olympic year, but this year is the year of the Olympics. And so these are 5 colloids that have synchronized for us to make the Olympic rings. So best wishes to the Tokyo Olympics. Thank you very much.
Thank you very much, Pietro. That was a really lovely talk. And it's really nice to see so many of the emergent behaviors, see them on this scale, and also the way you can generate them. So thank you very much. And we've just got time for a few questions, and then hopefully we'll get you back for the panel discussion. But one question is about scale. Is there any limitation on scaling these up? Does it have to work on the micron scale, or can we bring them kind of to the real world? Or what conditions are on these?
So the physics of all these systems does rely on low Reynolds number, which I should have stressed more. So there's no turbulence or there's no inertia. Everything is overdamped. But in that limit, people have done replicas up to larger scale. That's fine. At low scale, you have very interesting behavior that comes from the thermal fluctuations, so Brownian motion and noise, which biology has to cope with. And I suppose if we want to make systems that have large arrays, we maybe don't want to make it macroscopic. But I could imagine, I mean, the physics will hold macroscopic as long as it's low Reynolds number.
Brilliant, thank you very much. That makes sense. And one other question, you talked about the cilia in the brain. We're just wondering, do they actually play any role other than the mechanical role? Are they doing any computation or filtering within the brain, or is it similar to the process they perform in the airways?
So for sure, they're transporting nutrients. That's the sort of the textbook. Whether they're doing more, nobody knows. That science paper that I quoted is under a lot of discussion. It seemed to show that between the day and night, that there was a different flow pattern, which it could, at the very least, sort of have implications as an amplifier of a signal, or even as a switch. I think sort of creating one of these sort of or and switches is not that difficult. You just need sort of a few patches that end up having this diode behavior, and they will do that. So I think, but it's all to be explored. This is purely speculation, that the way I've answered.
Thank you. And final question. So the chemical computation is a really, really nice, exciting area, and to see these analogs in biological system. Based on your intuition, how far do you think it can go? Do you think we're going to see, could we see these as the brains of robots or the brains of systems? Or do you think, are there any fundamental limitations on the computational capabilities or power of chemical computation?
I think in the same way that sort of the quantum computer has been able to solve particular puzzles, I think people with sort of DNA arrangements have been able to solve their own very specialized puzzles in a very optimal way. But if we want a general purpose computer, well, it'll be tricky. But I think that the beauty of these sort of non-silicon approaches are that you can build them out of biology. You can have them fed by kind of some carbon fixation by a plant. That you might be able to just kind of make them self-assemble. They have, I don't think they're going to be as powerful as an Intel chip anytime soon, but they have other advantages. Thank you very much.
Yeah, that's really interesting. Finding the right niche for different types of computation, for different applications and different types of learning as well. Thank you very much. Thank you very much, Pietro. That was a really another fascinating talk.
Thank you,
And I'm sure there's more questions will come in and have come in on both the chat and this Discord. So hopefully we'll be able to catch up with those later and hopefully hear from you later as well. So thank you.
Thank you.
So in the next section of the workshop, we're going to move on to some panel discussions. So we're just going to have quick 10-minute talks by each of the speaker. And then we'll move on to a group discussion. So bring back all the panelists together and try and have a bit more of a discussion.